Thank you very much for the kind introduction. My name is Chen Yu, and I'll be introducing some of the microbiome applications from Novogene. Sorry, so please give me a second. Sorry. Hello. So, sorry. To kind of introduce myself, my name is Chen Yu, and sorry, can you see the next slide? Let me see. Sorry, please give me a moment. Okay, thank you. Sorry, my internet is a bit wonky. But to introduce myself, my name is Chen Yu, and you can also call me Valerie Joy Tan. And I actually received a Bachelor of Science from Calvin University with a concentration in biochemistry. My research experience during my bachelor was in development of HIV latency with a focus on host and pathogen interactions. And for my Master of Science, I received it from Illinois Institute of Technology with a concentration of biology for health professions. During this time, I received research experience in targeted cancer therapeutics and imaging with a focus on immunohistochemistry. So you can kind of see my research experience actually helped with my current work experience because I'm a current product manager for Novogene의. And the products I'm managing includes human-based solutions such as human whole genome sequencing and whole exome sequencing, as well as microbial solutions such as shotgun metagenomic sequencing, amplicon sequencing, and microbial whole genome sequencing. But without further ado, let me kind of introduce what we'll be discussing today. So for today, I will be introducing Novogene and then talking about some of the topics that's currently being researched in microbiome research and some of the sequencing applications within microbial applications. Then I'll touch briefly on the multi-omics that is currently emerging within microbiome research, and then sharing some of our team members. Please give me a second. Okay. So let me share about Novogene. Novogene is a leading provider of genomic services and solution with a cutting-edge NGS and bioinformatics expertise and the largest sequencing capacity in the world. So this is our mission. Our mission is to advance genomics and to improve life. For Novogene, our vision is to be the global leader in providing genomic services and solution. And our corporate value is on professionalism, innovation, integrity, and partnership. And as previously mentioned, we do have the largest sequencing capacity in the world. And so we actually have 10 laboratories and 18 subsidiaries around the world. Our headquarters is based in Beijing. So we actually do have a sequencing center in Beijing as well, known as Beijing Sequencing Center. But for Novogene의, the sequencing center that we have will be Singapore Sequencing Center at Novogene AIT Genomics, and have Novogene Genomics in Thailand, Novogene Hong Kong, and Novogene Japan at Kashiwa Todai Novogene Omics Core. So for samples coming from South Korea, the samples will actually be delivered to Singapore Sequencing Center. And because we actually have a lot of collaboration, we are co-published and acknowledged in around 22,000 SCI articles, and we have accumulated around 148,000 impact factor. For Novogene의 specifically, which includes the Asia Pacific, Middle East, and African region, we actually serve around over 10,000 projects annually from 15 countries. So from South Korea, we actually have Korean University, Seoul National University, Yonsei University, and Sungkyunkwan, University, and UNIST. And for Novogene, we actually have many different types of genomic services and solutions, which includes genomic, transcriptome, microbial, epigenomics, pre-made libraries, and metabolomics. For today, we'll be focusing on microbial. So for microbial, we have deep and shallow shotgun metagenomic, amplicon metagenomic sequencing, full-length 16 metagenomic, microbial de novo sequencing, and microbial whole genome sequencing. But before I dive into some of the applications that you will see in microbiome research, allow me to introduce microbiome research and some of the topics being covered. So microbiome research is actually defined by a microbiome. So microbiome, by definition, is microorganisms in a particular environment. Usually when we think about microbiome research, we think of human and animal-based microbiome research. Usually for human and animal-based microbiome research, we're looking and studying the microbes that live in and on the body and their roles in health and disease. Many of this type of targets include the gastrointestinal tract, so going from the mouth all the way to the colon and anus, as well as the skin surface, genital regions, and tumor microenvironment. In fact, tumor microenvironment is actually an emerging trend that's happening within microbiome research. Many of this type of studies are focused on host and pathogen interactions, diet or lifestyle such as smoking or other lifestyle changes such as exercise, as well as disease association, drugs and therapy response, bioengineering, and pathogen dissemination. But the topic of microbiome research is actually very complex because our microbiota actually interacts with our innate immune system. In fact, many of our digestion cannot occur without microbiota in our stomach. And so changes within our diet and our lifestyle can actually impact the composition of our microbiota. And this type of composition change can actually occur as quickly as 2 weeks. However, by definition, microbiome is looking at microorganisms in a particular environment. So microbiome research is actually not limited to human and animal-based research. Instead, there's actually a subset of microbiome research that's focused on environmental microbiome. Many of this type of environmental microbiome is looking at plants, soil, water bodies, which includes pond or ocean, wastewater, atmosphere, and material sciences. Many of this type of studies actually includes agriculture, bioremediation, and genomic surveillance of pathogens. An important thing to note about environmental microbiome is that they're often not done in isolation. So what this means is that environmental microbiome actually does play a role in human and animal-based microbiome as well. So if you look into how microbes are spread within the food system, you will see that it actually incorporates both human and animal-based, such as diseases or microbes on the food or in human environment, as well as human diseases. But it also incorporates environmental-based microbiome, such as the soil environment, plant environment, and aquatic environment. So oftentimes, environmental-based microbiome and human-animal-based microbiome are related to each other. And this is actually an ongoing trend as well. Actually, for example, in Nature, they actually published a news feature early this year where they're looking at antibiotic resistance and how it is growing in human communities as a result of global change, climate change. And this climate change is occurring because of a global warming, and this type of spread is actually causing flooding to occur. And by this type of flooding, you actually are taking microorganisms coming in from the environment and entering human spaces. And so they're actually seeing an increase in this type of antibiotic resistance. And so now let me introduce some of the sequencing applications that we do see within microbiome research. An important thing to note about our sequencing methods, both in Novogene and also seen within microbiome research, is that many of this type of studies or sequencing method is dependent on the sample type. The majority of microbiome research is actually done on mixed samples, or what we call as metagenome. So this type of samples are genetic information coming from a mixture of microorganisms. And so there are two main types of sequencing method for this type of mixed sample. The first method is through metagenomic sequencing, which includes shotgun metagenomic sequencing, whether it's deep shotgun or shallow shotgun metagenomic sequencing. This type of methods is typically done through short read sequencing. There is also a long read sequencing as well, and this is done through either PacBio or nanopore for metagenomic sequencing. The second method is through amplicon sequencing. So amplicon sequencing actually differs quite drastically in comparison to metagenomic sequencing. For amplicon sequencing, this is divided through either short reads and long reads as well. For short reads, we have 16S, 18S, and ITS amplicon sequencing, while for long reads, we have full-length 16S sequencing. Thus, after many researchers have finished looking into mixed samples, they will often isolate specific bacteria species or fungal species of interest. It is only after they have isolated and maybe even cultured those specific species of bacteria or fungal that they can actually move on to this other branch, which we're working with single bacteria or single fungal species. This type of sequencing method is divided by whether this species has a reference genome or no reference genome. For a single bacterial or single fungal species that do not have a reference genome, we typically provide de novo sequencing through long-read sequencing, either through bacterial complete mat or fungal find mat. And for single bacterial or single fungal species that do have a reference genome, this is typically done through microbial resequencing, either through short reads or long reads. But since the majority of microbiome is working with mixed samples, I would like to focus on metagenomic services. For metagenomics, this is defined by the genomic analysis of microbes by direct extraction and cloning of DNA from a microbial community. So metagenomics actually provides a culture-independent insight into microbial communities. And that's because usually many researchers are taking microorganisms straight from the environment of interest, whether it's human-based such as stool samples or environmental-based such as soil samples. And then they'll take this type of microorganisms and then they'll either use the whole metagenomic sequencing method through shotgun metagenomic sequencing or through amplicon sequencing. The general goal of this metagenomic services is actually to characterize the metagenome and to understand the potential functions of the microbes present within the sample. And as previously mentioned, I mentioned that the two sequencing method is actually very different from each other. For whole metagenomics, you're actually taking the total DNA from your sample. So you're working with the total metagenome and then sequencing that. As for amplicon sequencing, we're using specific primers to actually sequence a portion of the DNA of interest and sequencing that. And then I will be explaining more of them in detail. So the first one will be shotgun metagenomic sequencing. For shotgun metagenomic sequencing, I have previously mentioned that we are taking the total DNA from the sample and then fragmenting it and then sequencing it. So this actually allows researchers to be able to understand their samples with a lot of information, which includes species classification, as well as a better representation of their species in terms of proportion. But through some of the analysis methods they can do with shotgun metagenomic sequencing, you can actually identify gene function, metabolic pathway, and antibiotic resistance gene information from your sample as well. This means that for shotgun metagenomic sequencing, you can actually answer two questions that researchers are trying to answer. The first is who is there, and then the second is what are they doing? So for shotgun metagenomic sequencing, you can actually identify both the taxonomy and the function of your sample. And as I previously mentioned, the ability for shotgun metagenomic sequencing to look into antibiotic resistance genes is actually very important, especially in environmental microbiome, especially since that the spread of antimicrobial resistant microorganisms is on the rise. So this is actually a pretty hot topic in terms of environmental microbiome research. And for shotgun metagenomic sequencing, because we're fragmenting the DNA into smaller pieces, many of this sequencing method is done through short reads, either through Illumina NovaSeq 6000 or Illumina NovaSeq X Plus. However, you can also do this type of sequencing through long-read sequencing as well, either through PacBio or Nanopore. Then we have the second sequencing method, which is the amplicon sequencing. For amplicon sequencing, this is a different type of sequencing method and approach to looking into a metagenome. For amplicon sequencing, you will actually take specific primers that actually correspond to one specific diversity of microorganism. So for example, 16S will be looking into bacterial diversity, while ITS will be looking into fungal diversity. 18S includes eukaryotic microbial diversity, which includes both fungal and protist information. And these primers are actually very specific to the type of species that they can actually target. And how you do this is actually you take your total DNA, you use your specific primers, and then amplify those specific genetic information of interest. So in total, this method is known as metabar coding. And so amplicon sequencing is actually trying to answer a question of who is there and understand the composition of microorganisms present within the sample. And so you can see that amplicon metagenomic sequencing is actually very drastically different in comparison to shotgun metagenomic sequencing. However, a downside to amplicon sequencing is that this is kind of limited by the specific amplification primer that is chosen. So 16S full-length metagenomic sequencing is actually introduced to help resolve this potential bias when selecting one amplification region over another. That's because 16S full-length metagenomic sequencing is actually amplifying from the variable region 1 to the hypervariable region 9. So you're actually sequencing the entire 16S region, and you will actually result in an amplicon that is around 1,500 base pair. And this, because you have a longer amplicon size, so this type of sequencing is typically done through long-read sequencing on PacBio. So we actually offer a PacBio Reveal platform and PacBio Sequel 2 platform. The benefit of 16S full-length amplicon sequencing is that it does offer higher accuracy and resolution in comparison to short reads such as Illumina sequencing with 16S, because it can actually offer species-level resolution. And then another benefit of 16S full-length is that because there's no preference for a specific species, customers actually don't need to sequence multiple target regions in order to retest different regions. So this is another sequencing method that falls under amplicon sequencing as well, but through a long-read sequencing method. And that actually kind of concludes some of the metagenomics services. And now I'll be introducing the microbial whole genome sequencing. So microbial whole genome sequencing is typically done after customers have already identified the microorganisms of interest, and they will isolate and even culture specific species of interest. For microbial whole genome sequencing, this is actually divided into two methods of sequencing. And this is dependent on whether the specific species of interest has a reference genome or not. For species that do not have a reference genome, we actually have to do de novo sequencing. That's because de novo sequencing is actually the sequencing of a novel genome of a species. And the goal of de novo sequencing is actually to obtain this assembled genome. So because you're trying to obtain an assembled genome, you will actually need to assemble the sequence data. And this is very different in comparison to whole genome resequencing. Whole genome resequencing is done for species that do have a reference genome. And so for whole genome resequencing, the goal is actually to compare this species with the reference genome. So the goal is actually to look at genetic differences from the same species. So you're looking at genetic mutations such as SNP, indel, SV, and CMV. So these are two sequencing methods, and I'll touch briefly on both of them. For microbial de novo sequencing, because the goal is actually to obtain an assembled genome, this type of sequencing is typically done through long-read sequencing. That is because long-read sequencing actually provides more accurate assembly, and it can actually resolve repetitive elements within the microbial genome, and also actually improve the contiguity of genome assemblies. So that's why for microbial de novo sequencing, this is typically done through long-read sequencing, either through PacBio or nanopore. In comparison, for microbial resequencing is actually just sequencing the species of interest and trying to understand how the species is different in comparison to the reference genome. So this can be done with both short reads and long read sequencing. And the general goal is actually to compare the species sequence information with the reference genome to identify variants such as SNP, indel, CMV, and SV. And then now I'd like to share a little bit on multi-omics. So we are seeing a more multi-omics approach within microbiome studies. And what multi-omics means is that it's actually using an integrated approach to study complex biological systems and processes. What this means is that you're actually incorporating not only microbial solutions, but other solutions in order to understand the microbiome. So I'll be using a case study as an example. This case study is looking at the application of multi-omics within the gut and brain axis of a honeybee model. This case study actually incorporates two studies, and they actually use many sequencing method. So we actually incorporated shotgun metagenomic sequencing, proteomics, transcriptomics, and metabolomics. And it's trying to understand how the gut microbiome of the bee can actually impact the brain of the bee. And in general, they actually found that to have a conventional gut microbiome within the bee is actually necessary for the honeybee to cultivate both learning and memory abilities. And they actually identify specific bacterium of interest, such as lactobacillus apis as well. So this is actually an ongoing trend that we are seeing now for microbiome studies. And because of this, allow me to introduce that we do have other sequencing services in inclusion to microbiome sequencing services as well. So this actually includes genomic sequencing services for both human and plant animal, as well as transcriptomics such as metatranscriptome, mRNA sequencing, single cell and spatial transcriptomes. And then also metabolomics such as untargeted metabolomics and targeted metabolomics. And if you have any questions, please feel free to reach out to our team members, which includes Xinya and Xiao Rui. And you can actually reach them through their email address as listed here, as well as their QR code for their Kakao Talk as listed here as well. And that kind of brings the conclusion to this webinar information. Thank you very much. And please let me know if you have any questions.